All right, so welcome to lecture 3 of Biochemistry and Society. And so in this lecture, we're going to start talking about biological or organic building blocks. So these are specific chemicals that are going to be, in general terms, combined with each other like Legos or like building blocks to make the 4 classes of biological macromolecules that we talked about. So proteins, nucleic acids, lipids, and carbohydrates. So proteins are going to be polymers of amino acids. So those amino acids are going to be linked together in a specific order and have specific chemical characteristics that makes the overall structure and function of the protein. So we'll talk about amino acids, their different chemical characteristics, and how they're grouped together to make proteins. We'll also talk about nucleic acids, so the genetic storage system in the form of DNA, and the genetic conveying system, which is in the form of RNA, and also their role as energy molecules. We'll talk about the individual building blocks of nucleic acids like DNA and RNA, which are called nucleotides. So we'll go over the chemical structures of those. We'll also talk about fatty acids, which are sort of the building blocks of lipids. So lipids don't really form polymers, but they form aggregations, especially with cell membranes. And then we'll talk about carbohydrates, so simple sugars that'll actually combine to form these complex carbohydrates that you use in your body. So, and we'll talk about some interesting things along the way, little side steps. We'll talk about Coca-Cola and sperm whales, and sugarcane production and stuff like that. So it should be a pretty interesting lecture for you. So I hope you all enjoy it. Let's go ahead and get started. Lecture number 3, organic building blocks. So if you'll remember from our earlier lectures, there's 4 major classes of biomolecules. So there's proteins, nucleic acids, lipids, and carbohydrates. So generally, all of life's processes are performed by these large biomolecules. And then most of these biomolecules are polymers, except for lipids, although lipids do make associations. So really, if you're going to understand anything of biochemistry, you do need to understand that proteins, nucleic acids, lipids, and carbohydrates are responsible for basically all of life's processes. So since these molecules are basically made up of polymers, each of these molecules, proteins, nucleic acids, lipids, and carbohydrates, are made up of more simple organic building blocks. So these simple organic building blocks will then connect with each other, basically like Legos or something like that, to form these large biopolymers. So these proteins, nucleic acids, lipids to some extent, and complex carbohydrates. So for proteins, they're made up of polymers of these chemicals called amino acids. So for nucleic acids like DNA and RNA, so nucleic acids, they are going to be made up of polymers of this type of chemical right here, which is called a nucleotide. Now, lipids are a little bit more complicated, but in general, lipids are either these fat molecules or they're made up of fat molecules, which are depicted right here. And then complex carbohydrates, which basically means carbohydrate polymers, are made up of individual sugar molecules like shown right here. So you can already see that amino acids, nucleotides, fats, and sugars have very different sort of chemical structures. So we already talked a little bit about amino acids. So they all have this region in common with each other. Nucleotides are a bit more common or are more complex. So this nucleotides are always made up of a nitrogenous base. So this molecule right here that contains these nitrogen molecules, and it forms this ring structure. Then they also actually have a sugar molecule, a ribose sugar or a deoxyribose sugar, and then they have these phosphate molecules here. So nucleotides all have that general basic structure. Same with fats that make up lipids. So fats in their most simple form have a carboxylic acid group right here, and then this long hydrocarbon chain. And then sugars also have this very unique structure, which you can see right here. And sugars are basically carbon hydrates. Carbohydrates come from that. So hydrate basically means hydrogen and oxygen. And so sugars are made up of repeating units of CH2O. But you can basically just think of them as a bunch of carbon molecules attached to oxygen molecules, as you can see in this structure right here. So these are the four most important building blocks of those different classes of biomolecules. So proteins are polymers of amino acids. Nucleic acids like DNA and RNA are polymers of nucleotides. Fats or lipids are basically a little bit more complicated, but they're all made up of these type of fat molecules. And then carbohydrates, long carbohydrate polymers, are made up of basically polymers of these individual sugar molecules. So we'll be talking about these building blocks of the biomolecules today. And then the next lecture, we'll go into more detail into all of the main biomolecules, the polymer forms. So because proteins are so important to biochemistry, we can actually start this off by talking about amino acids. So amino acids are the building blocks for all proteins. So amino acids are just specific types of chemicals that have a shared characteristic. So all amino acids have this amino group here, which is a nitrogen containing group bounded to a carboxylate group or a carboxylic acid group. And they all have a hydrogen here. And all three of these groups are connected to a central carbon, also called the alpha carbon. So all amino acids have this group. So all amino acids have these groups. Now, all amino acids also have another group bound to that central carbon. And so this is called the R group, which just means any group, or the side chain. So this group is different for each of the amino acids. So all the amino acids have that central alpha carbon, the amino group, the carboxylic acid group, and then a hydrogen. And then they're all bound to an R group, which could be any of the different, well, actually 20 different chemical groups. So that's how you get the different amino acids. You basically put different chemistry onto that in place of that R group. Now, an interesting thing about amino acids and other molecules in chemistry, overall, is that when a carbon atom is attached to four different groups, as we have with amino acids, so amino acids are the carbon attached to the hydrogen, the amino group, carboxylic acid group, and an R group, and it's going to form this tetrahedral form, which we talked about in the last lecture. So when a carbon is bound to four different groups, that molecule can actually be right or left-handed, depending on how those groups are related to each other. So we have one molecule right over here, and this is called the L isomer because it's basically left-handed. But then if you arrange these groups differently, you can get the D isomer, which is basically right-handed. So you can see this one is also attached to the four different groups. However, the position of this group and this group are different between these two molecules. So they're said to have chirality. So they're basically mirror images of each other, but they're not able to superimpose on top of each other. So if you take your hands, your right and left hand, and you put them next to each other, you can see that they form mirror images. But if you try to put one hand on top of the other, and you can just try that right now, you'll see that they don't superimpose. The same thing happens with chemicals that are attached to four different groups. Now, for some reason, all the amino acids on all life of Earth that have ever been found in a biological system are left-handed. So all amino acids in all of life on Earth are in the L isomer. So why this is the case, nobody knows. The reason for this is completely lost to evolutionary history. So we know that humans all have L amino acids, but we do not know if some alien species or life on another planet might have D amino acids. So that's just a little interesting tidbit about amino acid chemistry. So basically, all of life, so all proteins and all living things, for the most part, are made up of polymers of these 20 different amino acids. And they're all shown on this slide right here, all 20 of them. So the reason why these specific 20 are incorporated into biological systems is not known. There are a lot of other amino acids that exist. There's many other amino acids that are doing sort of functions like metabolic functions in your body, but all proteins in all living things are pretty much just made up of these 20 amino acids. What's interesting about this is that you remember that R group that is variable between different amino acids. So each of the different amino acids will have a different R group attached to that central carbon. You can see some are very simple, some are very complex, and they have different shapes, they have different chemical characteristics. You can see this one has an aromatic ring, this one also has an aromatic ring, other ones don't. This one's got an oxygen. This one's got an oxygen in another group. This one's got an aromatic group and an oxygen. This one's got a sulfur. This one's got this complex carbohydrate group. This one's got this group right here. I'm just going to go through them all now that I started. This one's positively charged. So is this one. And these two are negatively charged. These are all 20 of the different amino acids. And so each of them has a different chemical characteristic on that R group. Some of them are hydrophobic, some of them are polar and able to make hydrogen bonding interactions with water or other molecules. Some of them are positively charged and could maybe interact with a negatively charged ion. They all have a different chemical characteristic. Now, if you can understand this, you can sort of understand how proteins work. So proteins are going to be polymers of different amino acids folded into a different three-dimensional shape. Now, the types of amino acids that are on that specific protein are what's going to cause it to fold in a specific way, and it's going to cause it to do its function, its biochemical function within the cell. So the fact that there's 20 different amino acids and they all have different chemical characteristics is really important to understanding proteins and biochemistry in general. So each of these amino acids has different properties, but they can be broken up into 5 different groups, basically, based on their shared characteristics. And that's what I've sort of done here. So in this box right here, there's 7 amino acids that are hydrophobic, have hydrophobic side chains. Basically, these side chains don't want to interact with water. And they're aliphatic, which that's not a term you really need to know. It's basically they're just made up of these hydrocarbons for the most part, and with no aromatic rings. So you see this one just has a hydrogen, so that's hydrophobic. This one just has a methyl group, that's hydrophobic. This one has it's just basically a hydrocarbon chain, so it's not going to interact with water. Same with this one, same with this one. This one just has hydrocarbons, even though it forms this ring. And this one has a sulfur atom, but basically it just has these groups that are hydrophobic. Now, there's also 2 amino acids that are hydrophobic, but they have these aromatic rings. So you can see right here, there's no oxygen or anything that can interact with water, but these are basically hydrophobic, but they have aromatic rings. So this is another interesting one right here. There's then 6 amino acids that have polar side chains but don't carry a positive or negative charge. So you can see this one has a methyl group and an oxygen, a hydroxyl group or an alcohol group. So that oxygen atom, of course, is going to be electronegative, and so that changes the charge a bit so it can interact with other molecules. So it has a polar side chain. This one also has an oxygen group. This one right here has a hydroxyl group on top of this aromatic ring. This one has a sulfur group, which you know from our previous lecture behaves very much like alcohol, but a little bit differently. Then this one has what's called both of these actually have what's called amide groups, which is a carbon double bonded to an oxygen and double bonded or single bonded to an amino group. So these are all polar amino acids, and they can interact with other stuff. So then down here we have 3 amino acids that carry a positive charge. So this one right here, you say it has an amino group that has a positive charge. And this is basically at pH 7, it'll have a positive charge. And then there's this guy right here, which sometimes can carry a positive charge. And then there's 2 amino acids over here that carry a negative charge at pH 7. So you can see, you can kind of break these amino acids and their chemistry up into different groups. There's hydrophobic, aromatic, polar, positively charged, and negatively charged. So if we look a little bit more specifically at these amino acids, and by the way, you do not need to remember any of these amino acids. If you want to, that's great, very fun, but obviously there's no tests where you're going to be have to remember this information. If you ever were to become a biochemistry major and take a biochemistry, you will eventually have to learn the names of all these different amino acids. But these are the 7 amino acids that are hydrophobic and aliphatic. So the first one is this one called glycine. And so it's the simplest amino acid. It actually doesn't have chirality because it's not attached to 4 different things. The R group is just another hydrogen. So each amino acid has a full name, a 3-letter code, and a 1-letter code. So I'm not going to go through all these because you don't need to know that. But it's glycine, gly, and G. So when you're saying a protein sequence, you can use these codes and sort of just do your protein like this, and you know that's a glycine, a methionine, an alanine, a threonine, a cysteine, and a glutamate. But anyway, glycine's the simplest amino acid. The second most simple is alanine. That just has a methyl group right there. And then you've got 3 what are called branched chain amino acids, and those are actually getting pretty popular in like the nutrient field. So that's another CH3. And so these have just hydrocarbons, but they have a little bit different shapes. So these can sort of fit together like puzzle pieces in the inside of a protein. You also have methionine. So this one's got 1, 2, 3 methyl groups and attached by a sulfur, but it's still hydrophobic for the most part. And then you have this interesting one here, proline, and that's the only one where it actually forms a ring where it sort of comes back and bonds with this the amino group on the amino acid. So these are the 7 amino acids that are hydrophobic and called aliphatic because they're just made out of, for the most part, hydrocarbon chains with no aromatic structures. So there's 2 amino acids that are hydrophobic but aromatic. So one is phenylalanine, shown right here. So this is the side chain. So it's got just the CH2, and then it's got a benzyl ring right here. So that's called phenylalanine. And the other one you might have heard of is called tryptophan. And so it has the same thing, a methyl group, but then it's got this thing called an indole, which is a sort of complicated benzyl ring attached to a 5-membered ring that contains a nitrogen. So this is tryptophan. As I said, there's also 6 amino acids that are polar, but don't actually have a full positive or negative charge at pH 7. And so there's 6 of these. So one is serine. It has a methyl group and then a hydroxyl group. There's threonine, which is very similar, methyl group, hydroxyl group, but then it's got this other methyl group, carbon group sticking out. There's tyrosine, which is actually aromatic. It's just like phenylalanine, and it's actually derived from phenylalanine, but it has this OH group here that can interact with water and is also polar. We have cysteine, which is shown right here. It's just like serine up here, except the OH is an SH, so contains sulfur in it. There's asparagine, which is shown right here, which has this interesting amide bond, so it's a C double bond O and bonded to NH3. And then there's glutamine, which is the same as asparagine, except it's got 2 of these methyl groups instead of 1. So those are the amino acids that are polar but uncharged. Again, you don't need to remember them. I'm just introducing the different flavors to you. Now, there's also 3 amino acids that are positively charged at pH 7 for the most part. So these can interact with water, and they can also interact with other molecules that carry a negative charge or a partial negative charge. So one is lysine, which is shown here. So it actually can interact with other hydrophobic molecules from these methyl groups, but it's got a charged amino group at the top of that chain. And then there's arginine, which is kind of similar. It's got these ring of methyl groups here, and then it's got this group here, which is called a guanidino group. I think this chemical is found a lot in like bat guano or bird guano. But that can carry a positive charge as well because it's sharing sort of these double bonds between these different molecules. And then we have histidine here, which is actually really important for enzyme function because it can also can also be positively charged. So that NH group can gain a proton, and then it'll have a positive charge because this nitrogen is sort of partially charged to 4 things instead of 3 things. So this is histidine. And because it can sort of gain and lose this proton, it's really important for enzyme activity. So that's proteins that catalyze a specific chemical reaction in your cell. So those are the 3 amino acids that are positively charged. And then there's 2 amino acids that are negatively charged. So here's aspartate right here. And so it has this carboxylic acid group. It's got 1 methyl group and then this carboxylic acid group that can lose a proton and form this COO. That double bond is shared between these 2 oxygens, and it carries a negative charge. The other one is glutamate, which is just like aspartate, except it has 2 methyl groups instead of 1. So that's a little bit of a closer look at the 20 different amino acids that make up proteins. So humans are actually not able to synthesize all 20 of the amino acids in our bodies. Some of them we can, and those are called non-essential amino acids. So non-essential amino acids are listed right here. So alanine, arginine, asparagine, aspartate, cysteine, glutamate, glutamine, glycine, proline, serine, and tyrosine. Our body can take other molecules and modify them through, enzymatic chemistry and then turn those into these amino acids. So you don't have to eat arginine molecules to use arginine in your proteins. Now, the essential amino acids, so histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine, those are required in your diet. If you don't get enough of these amino acids, you'll develop basically a deficiency, and it can cause all sorts of problems because you need all 20 of these amino acids to build up every one of the proteins in your body. So if someone says, this contains essential amino acids, that means that those amino acids cannot be synthesized by your body. They need to be ingested through your diet. So now I'll just talk a little bit more about a few of the amino acids that are a bit more interesting and you might find interesting as well. So one of these is phenylalanine. So one thing that's interesting about it, so it's shown right here. So it's got this stuff. So basically right here is the R group that we were talking about. So the R group. And so it just has a single carbon methyl group, and then it has this benzyl ring right here. So phenylalanine is really important because it's modified. Apart from it being a part of proteins, it's also a chemical that's just modified to form other molecules. So that includes the other amino acid tyrosine. So remember that I mentioned that this amino acid is one of those polar amino acids, and it's just like phenylalanine, except it has this OH group right here. So you actually need to ingest phenylalanine in order to synthesize tyrosine in your body. So tyrosine is sort of on the bubble as to whether it's considered essential or non-essential amino acid. But you see, for phenylalanine, you just take it, add an OH group, and then you have tyrosine. So other things you can do is you can modify phenylalanine, and our bodies do this, to form neurotransmitters like dopamine. So dopamine, you can see, has a very similar sort of shape. This group right here is the same. This group right here is the same. And then this carbon here is the same. And then that's the same. So basically, you're cutting off this part of the molecule, and that's actually CO2. You can release it like that. And then you add these OH groups to these 2 parts on the molecule. I think actually it first starts as tyrosine and then gets converted into dopamine. And then you have dopamine, which is a really important neurotransmitter for your brain development, not just development, just normal function in your brain. So dopamine, if it's not functioning or if you have a deficiency, that can cause all sorts of problems. Another one is epinephrine. So this is a hormone involved in the fight or flight response. So like if you're walking down the road and you see a lion and you need to get away really quickly, your body's going to be flooded with epinephrine, which is going to tell all your cells to, stop digesting food and start, your blood vessels dilate, and it'll basically allow you to have a fighting chance to survive a stressful situation. So epinephrine, you can see, is also very similar to tyrosine, and that's because it's actually synthesized from tyrosine, or I'm sorry, from phenylalanine in your body. So that's interesting. Amino acid number 1, phenylalanine, and also we talked a bit about tyrosine. So another molecule that phenylalanine is converted into, which actually goes into our satiety discussion in this course, is melanin. So melanin, of course, there's a lot of different kinds of melanin, actually, but the one that you're most familiar with would be the pigment form of melanin, which is called eumelanin. So this is eumelanin. So eumelanin is this sort of complex structure, and you can have all sorts of groups coming off over there. But you can see it's actually synthesized from phenylalanine. So it's a little bit hard to see, but basically all these benzyl rings and stuff in this molecule come from phenylalanine and form these polymers that'll form eumelanin. So eumelanin, of course, it's produced in cells called melanocytes, and it protects skin from UV light damage. So you know that people who grew up or who basically societies that evolved in more northerly environments have less melanin generally in their skin. Actually, that's related to vitamin D nutrients. So that comes from being exposed to light. That actually allows your body to synthesize vitamin D. So if you have less melanin, you're letting in more UV light to allow you to make more vitamin D. Drawback of this, of course, is that UV light can damage your DNA in your cells and cause mutations that can turn into cancers and stuff like that. So it's sort of a give and take. And then pheomelanin is a related molecule as well that incorporates a different amino acid, cysteine. So it's in addition to these benzyl rings that come from phenylalanine, it also has these sulfur groups that come from the amino acid cysteine. And so pheomelanin is actually a compound responsible for red hair in people, and like this guy Rupert Grint right here. So and so it's involved in that. And of course, there's a lot of societal issues, obviously, surrounding melanin, but it's a very actually not a big genetic difference, the color of your skin at all. But there are issues with people who suffer from albinism, which is part of which is the inability to synthesize melanin in your skin. So there's a lot of societies around even today where people who suffer from albinism, there's a person right here, are actually ostracized and are accepted less by society in general. And that's caused all sorts of terrible horror stories in certain places where they can be exploited or even murdered because of the fear around their skin color. But really, it's just a chemical that's, important in protecting our skin from UV light. And obviously, there's been variation among human populations in the extent to which your melanocytes produce melanin. So everyone on the planet basically has the same number of melanocytes, but different societies in different geographical regions have evolved. Their melanocytes basically have evolved to produce more or less melanin. Another societal issue that sort of involves phenylalanine is this disease called phenylketonuria. And so this is actually an inherited metabolic disease where individuals are unable to do this modification of phenylalanine to tyrosine. So this is performed by a specific enzyme. I think it's phenylalanine hydroxylase, but it converts in protein or just the amino acid. So it'll add this OH group that we saw. So that's a specific enzyme that's going to do that. So if you have a mutation in that enzyme that you've inherited, you can develop this disease called phenylketonuria. And basically, you're unable to synthesize tyrosine. So two things happens. One is that your proteins can't function properly because you don't have tyrosine in them. And also, you're building up a bunch of phenylalanine in your body. So these can cause really, really bad problems, including developmental disorders, mental disorders, and stuff like that. So this mutation is inherited from the parents of children. So I'll talk a little bit about this in terms of genetics, but basically you have a dominant gene that's the correct form of this protein right here that converts phenylalanine to tyrosine. However, you could also inherit from your parents a recessive mutated gene where this protein is ineffective. Now, in the father here in this and in the mother, they're both called unaffected but carriers. Because they have one copy of the gene that's functioning properly, you wouldn't even know unless you did genetic testing that they were carrying a recessive mutated form of the disease. Now, the problem that occurs when two carriers have kids, you have a 1 in 4 chance that your kid is going to be completely unaffected and not a carrier because they inherited both the dominant correct forms of the protein. There's a 1 in 2, there's a 50-50 chance that you will be unaffected. You'll get the positive, you'll get the good gene, but you'll also get a copy of the recessive mutated gene. So that would make a 50% chance that the children of two unaffected carriers would themselves be unaffected carriers. Now, unfortunately, there's also a 1 in 4 chance that the child will just be affected, that they will inherit both of the genes for this protein that do not function properly. So in this individual, they would develop PKU and not be able to modify phenylalanine to tyrosine, causing all sorts of problems. Now, society wonderfully has developed ways to combat this, and it's basically through genetic newborn screening. So as soon as a baby is born, and in this day and age, in this country and a lot of other countries, that baby is going to be tested for a panel of different genetic diseases that they may inherit, and that can, a lot of times, I think a lot of them can be treatable. So if you can tell right after a child is born that they are basically going to be affected by this disease, then what you can do is basically modify the diet of the baby through childhood, through everything, and do other sorts of treatments. And then someone with phenylketonuria, if you have that modified diet, they can live a completely normal and healthy life with the one exception that they have to sort of watch how much phenylalanine they're ingesting and make sure that they ingest a lot of tyrosine. So that's phenylketonuria. It allows us to talk about genetic inheritance and newborn screening methods that are in place in society. So another interesting amino acid is another one of these hydrophobic aromatic ones, and it's called tryptophan. So tryptophan has kind of the bulkiest side group of all the amino acids. It's this really big group here. You've got a methyl group, and then you have this indole ring that's made out of this benzyl ring, and then this 5-membered ring that has a nitrogen group right there. So tryptophan's also a pretty cool molecule. One reason why it's very interesting is that the amino acid is converted into serotonin, which is really important neurotransmitter. You can see that these two structures are pretty similar. So this indole ring in serotonin is just going to be derived from tryptophan. So another molecule that tryptophan is converted into is melatonin. So melatonin, you can see it also has this indole ring here, and that's because it's a modified version of tryptophan. And so melatonin, you may have taken like sleep supplements or something. So melatonin's really important in regulating our circadian rhythms, allowing us to basically get sleep. So melatonin levels will rise when it's bedtime, and then in the morning they'll decrease. Now, one thing that about tryptophan, it is the bulkiest amino acid, and it is kind of shaped like a turkey in at least the way I'm looking at it. But despite sort of popular folklore, tryptophan is not present in more of concentration in turkey meat than any other meat. So the meats, obviously, meat, turkey, pork, cattle, or beef, or whatever, is going to be made up of proteins, of course, but those proteins are going to have basically roughly similar numbers of each of the different 20 amino acids. That gets a little complicated, but regardless, there really isn't a huge link between the amount of tryptophan in turkey. So that was always said that's why it made people sleepy, because people figured that, oh, well, tryptophan is turned into melatonin. And so when you eat a lot of turkey, because there's a lot of turkey in there, it puts you to sleep. That does not seem to be the case. So it hasn't been studied extensively, but basically what is responsible for this sort of sleepiness after meals is just thought to be a high level of carbohydrates in that you're eating. So those carbohydrates can actually cause you to actually retain more tryptophan in your blood, which can be turned into melatonin. So the details of that have not been worked out, but there's definitely is a link between overeating and getting sleepy. But the whole myth about tryptophan being that eating turkey, it has a lot of tryptophan that makes you go to sleep, is not a thing that holds up on closer inspection. So there's another interesting amino acid, or that at least is one of my favorites, tyrosine. That's the other amino acid that contains the aromatic sort of side chain. So it's an aromatic ring, just like phenylalanine, but it's got this hydroxyl group here. Now this one's just sort of cool biochemically, and when I'm working on protein structures, I always get a little smile when I see tyrosine, and that's one of the reasons for that is that if you look at tyrosine, it sort of has this region right here that's hydrophobic, and then it's got this region right here that's hydrophilic. So tyrosine can do this cool thing in cell membranes, which are hydrophobic environments, where this part would be sort of embedded into the membrane, and then this OH group will sort of stick up. So tyrosine can actually sort of snorkel in a cell membrane. So this part right here would correspond to that part right here, and then this part right here would correspond to that OH group that can actually interact with water outside of the membrane. So that's just kind of a cool chemical feature that I like to point out about tyrosine. So the next amino acid I'd like to mention is asparagine. So this was actually, which it's shown right here, and so its side chain looks like this. It has that amide group where it's a C double bonded to an oxygen and a single bonded to a nitrogen. This was actually the first amino acid that was ever isolated and characterized. So it was done by French chemists in 1806, and it was isolated from asparagus juice. So there's a lot of asparagine in asparagus juice, and that's how it got its name. So just want to point out that I've heard it said that this isn't really the reason why asparagus makes your pee smell funny. That's actually due to this compound right here. So it's a sulfur-containing compound, which is where you get that strange smell, and it's called asparagusic acid. And then the last interesting amino acid that I would like to mention is glutamate. So glutamate is, maybe my favorite because I like food a lot, and that's because glutamate is basically responsible for umami. So umami is a Japanese word because they were the first one to describe it. And it's a Japanese word that basically means pleasant, savory taste. So it's sort of the fifth taste that you can get. So if you're eating meat or stuff like soy sauce or tomatoes and stuff like that, it's what gives those foods sort of a rich, meaty type of taste. And that comes from this glutamate amino acid right here. So it has this side chain here. It's actually negatively charged at pH 7. But it has this carboxylic acid group. So the link between glutamate and umami was first discovered by this guy, Kikune Ikeda, in 1908. And like I said, it's present in savory foods like meats and soy sauce and tomatoes. So actually the additive MSG, which you might have heard of and is used in a lot of different soups, very famously in Chinese recipes in America and elsewhere, I assume, MSG stands for monosodium glutamate. So it's just a glutamate salt. So there's a lot of people who sort of freak out about MSG and say, oh, I have this terrible reaction. Maybe they are. That has not been concluded to a high degree of evidence yet. And it is related because glutamate is a neuroreceptor or a neurotransmitter. So they're like, oh, I ate a bunch of MSG. It's sort of messing with my brain. So it's one of the main excitatory neurotransmitter. I believe GABA is the main inhibitory neurotransmitter. So this reaction to MSG was called a lot of times Chinese restaurant syndrome, which really doesn't make any sense. So Chinese food in America and probably elsewhere does often contain a lot of MSG, but so does Italian food. So there was no really good reason calling it Chinese restaurant syndrome as opposed to Italian restaurant syndrome or some other type of restaurant syndrome. But anyway, so that's glutamate. It's amino acid and it's an MSG, and it's wonderful because it makes everything taste really, really yummy. So now that we've gone through the different amino acids, the 20 different amino acids, we can sort of talk about how they come together to form these amino acid polymers that we know as proteins. So the 20 different amino acids in any number of combinations can come together, and they basically form what's called these peptide bonds. And so these peptide bonds are shown here. So this amino group over here, and you have the carboxylic acid group. The carboxylic acid group can interact with the amino group of a different amino acid, and then you lose a water molecule. And now these two amino acids are bonded together by a covalent bond, a very strong covalent bond. And you can see this can be continued for many, many amino acids. You can have amino acid, what are called polypeptide chains, and that's because these amino acids are linked by these peptide bonds. That's the type of bond that they're called. You can have proteins that are very small. You can have peptides that are just this big, just 5 amino acids, or some that are many, many thousands of amino acids long. And so these amino acids, they're always written from this amino terminus right here to the carboxy terminus, and that's because that's how they form. So in your body, these peptides are being formed like 1, 2, 3, 4, 5. And so those are formed in that way, and they're also written in that way. So if you were to write out this amino acid, you could write it like that. You could also use the one-letter code. So Y is tyrosine, G is glycine, G is glycine, F is phenylalanine, and then L is leucine. So if you wrote it like this, you would just assume that it was written from the amino terminus to the carboxy terminus end. So that's how amino acids will form these polypeptide chains to make proteins. And so we're going to talk about proteins more in the next lecture, but these amino acids form proteins, and that the formation of basically these peptide bonds between these 20 different amino acids in a particular sequence, that's referred to as primary structure. So that's just the specific order that these 20 different amino acids are in this protein. And you can see this particular one has a proline, then a lysine, then a valine, then a lysine, then an alanine, then a histidine, then a glycine, then a lysine, and so on and so forth. Now, these individual amino acids in the chain will interact with neighboring amino acids. So amino acids just a few parts down to form what's called secondary structure. And so these will actually fold into these really stable forms like this one right here called an alpha helix. So these backbones on the amino acids will actually interact with each other to form this sort of helical sort of thing. And it's really stable because these bulky side chains are going to stick out from the side of these like this alpha helix. There's another one too called a beta sheet. So we'll talk more about that next time. But basically, because these side chains stick off the side here, these side chains can interact with other side chains on other parts of the protein to make a full folded protein. So a full folded polypeptide chain in 3-dimensional space. And that's called tertiary structure. And then to add even a little bit more complexity on it, this tertiary structure, multiple polypeptide chains can come and interact to form a fully functional protein. And that's called quaternary structure, when more than one polypeptide chain that's already folded comes together to make a fully functional protein. So this protein right here is hemoglobin, and that's responsible for carrying oxygen around your body. And it's actually made from 4 different polypeptide chains. So 2 are the same, 2 are different. And it does not function correctly unless all of those different folded polypeptide chains or proteins are present. So we'll talk a little bit more in the next lecture about proteins more in general, now that you've seen how they're built up from these individual building blocks called amino acids. Now, the next class of molecules that we're going to talk about are nucleic acids. And so these are chemicals like DNA and RNA that are built up from these molecules, actually called nucleotides. And so nucleotides have a very characteristic chemical structure that basically comes in 3 parts. So the first part is right here. And so that's called a nitrogenous base. And these are just specific molecules that have these cyclic rings and they have a bunch of nitrogens in them. So these are called nitrogenous bases. So if you think about DNA, if you've ever heard like there's A, T, C, and G, the A, T, C, and G are just different nitrogenous bases that are going to be at this position. Now in the second position is this right here, or the second feature is this right here. And so that's actually a sugar molecule called either ribose or deoxyribose. That's the main difference between DNA and RNA. And so this is just a sugar ring that links that nitrogenous base to the third part of the molecule, which is 1 to 3 phosphate groups. And so these phosphate groups are really important. They're fundamental to form the polymers in DNA and RNA. So the backbone, the thing that's connecting those polymers, are actually one of these phosphate groups. And then it's also really important for providing energy. So this molecule here, ATP, this specific chemical that you're looking at, you can actually remove this phosphate group, and that provides energy that can be used by enzymes to actually catalyze chemical reactions that otherwise aren't thermodynamically efficient enough to occur. So these are really, nucleic acids and nucleotides are really important chemicals. So as I said, these nitrogenous bases, sometimes just called bases or nucleobases, these are specific nitrogen-containing molecules that have either a 2-membered ring or a 1-membered ring, but both of these rings contain multiple nitrogen groups on them. So these can be categorized in 2 different ways. The 2-membered rings are called purines, and so that's right here. And then the 1-membered rings are called pyrimidines. You don't need to remember those names or anything. That's just the 2 different types of nitrogenous bases that are found in DNA and RNA. And so, and then there's 5 different types of nucleobases or nitrogenous bases that are important. So one is adenine, and that can be just shortened to A. Guanine, which should be shortened to G. Adenine and guanine are both purines. They both have these 2-membered rings. And then there's 3 molecules that are pyrimidines, nitrogenous bases. So one is cytosine, which can just be shortened to C. And then there's thymine, which can be shortened to T. So in DNA, you can have A, G, C, or T. But then in RNA, this T is replaced with this uracil molecule, which is a U. And so that's found in RNA only, and then T is found in DNA only. So these are the different nitrogenous bases that are incorporated into the nucleic acids, DNA and RNA. So as I mentioned before, those nitrogenous bases are actually linked to sugar groups, so either ribose or deoxyribose. So that base is going to be linked at this position right here. And then the phosphate groups are going to be on this position of the sugar. So there's ribose, and so that is found in RNA. That's the sugar that's found in RNA. So it has this OH group here at the 2 position of this carbohydrate sugar. And then there's deoxyribose. So that's where we get DNA. So RNA actually stands for ribonucleic acid, and DNA stands for deoxyribonucleic acid. So that's a big difference between the 2 of them. So it does not have, deoxyribose does not have that OH group there. So nucleic acids, this base is connected to one of 2 of these sugars, which is then connected to the phosphate groups to make the nucleotide. And so this is not important for you to remember. This would be something for a more general biochemistry class, but we have the base here, and that could be called a nucleobase or just a base. Then if you take the base plus the sugar, that would be called a nucleoside. And then you have the base plus the sugar plus the phosphate groups, and that's a nucleotide. So if you only remember one of those, just remember nucleotide. But I just wanted to point that out. You really don't need to know too much about that. And as I mentioned before, nucleotides are really important energy molecules, and we'll talk more about this later in the course. But this one specific nucleotide called adenosine triphosphate, or ATP, that is the universal energy currency of all biological systems. So all biological systems will take in energy either through eating carbon-based molecules and oxidizing those, or through photosynthesis, and they'll use that energy to make ATP molecules. So ATP molecules is the universal energy currency because it provides energy, because if you break off this bond here, or even more of the phosphate bonds, you're left with ADP, adenosine diphosphate, and then energy is released when you break that bond. And it's the universal energy currency because every living system, from elephants down to the smallest bacteria, use ATP as their energy source. So why this is ATP and not some other nucleotide, so not the G, the C, the U, or the T, those ones, when you break their phosphate bonds, they release just as much energy as ATP. So why ATP is the universal energy currency is completely unknown and lost to evolutionary biological history. So another thing that nucleotides do is they can actually act as signaling molecules. So we'll talk a little bit about this in lecture 9, I believe, 8 or 9. But they're really important in signal transduction pathways. So signal transduction or cell signaling is just how your cells basically in a multicellular organism communicate with each other. So the fight or flight response is a really important one in that. So like if you're walking down the street and you see a lion, your body floods your bloodstream basically with epinephrine. That binds to cells, and then they'll start making molecules like this that'll change, which is a nucleotide called cyclic AMP, and it will completely change the environment of the cell and prepare it and other cells to sort of have a response to allow you to get away from that lion. These signaling pathways are also important for things like cell growth. So this is a protein RAS here, and it's actually bound to a nucleotide. And so that nucleotide is actually going to regulate its activity. Now, proteins like this, like RAS, are actually often involved in cancers. So if your cell gets a signal that it needs to replicate because you had an injury or something like that, that's great. But sometimes there's mutations in these types of proteins that can actually cause cells to proliferate or replicate uncontrollably, and that can lead to tumors. And then if other biological issues happen, it can lead to metastatic cancers. So nucleotides are also really important signaling molecules. And nucleotides also function as really important what's called cofactors. So these are molecules that your body uses to do different jobs in concurrence with proteins. So I'm not going to go into a lot of detail on this at all. But when we talk about metabolism, we'll talk about these two molecules, FAD and NADH. So they both contain nucleotide portions on their structure, as you can see right here. And these are really important for metabolism. So these molecules will actually take electrons from these carbon-based molecules that you ingest, like glucose, and then transfer them so they can be used, the energy in those electrons can be used to make ATP. So those are really important. Another really important one is this coenzyme A. So it's obviously it's A, so it's really important, but it contains a nucleotide here. And this is involved in metabolism as well because it carries these two carbon groups from place to place in your cell. So sometimes part of metabolism happens outside of a mitochondria and it's got to get into the mitochondria, or move around within the mitochondria. So coenzyme A is really important for that. So nucleotides are also really important cofactors, particularly when we're talking about metabolism. So these nucleotides are actually the building block, as I've mentioned before, for DNA and RNA. So DNA and RNA are formed from oftentimes very long polymers of these nucleic acids. So each of these is a nucleic acid. So the base is just written as base right here, and here's the ribose sugar, and then there's a phosphate group. So what'll happen is this phosphate group, it'll lose those two extra phosphates and then link onto the ribose sugar of another base. And so on and so forth. And then you get these very long, oftentimes, molecules. So the DNA in your body, it forms, well, it's broken up into different chromosomes, but you basically have, I think it's 3 billion bases of DNA in your body. And so these form very, very long polymers, many millions or billions of bases long. RNA is usually smaller, but it also forms these really long polymers. So there's only a couple differences between DNA and RNA. They do differ in their function, and we'll talk more about that in a bit. But DNA, it only has this deoxyribose sugars, so there's no OH group, whereas RNA has these ribose sugars, as I mentioned earlier. DNA has ACG and T bases, whereas RNA has A, C, G, and U. And then DNA usually forms a double-stranded helix, which we'll talk about, and which you've probably seen before. Whereas RNA, and this has a lot to do with their different functions as well, that usually remains single-stranded. So the difference between DNA and RNA in their function is that DNA basically stores genetic information, whereas RNA conveys that genetic information. So the genetic information is stored in the form of DNA. So the DNA in your cells was inherited directly from your parents, and it would be passed on to any children that you might have. So it's a complete record that holds the history of the sexual reproduction in which that DNA derived from. And it really is the engine of evolution, because mutations in DNA are going to be pretty much stuck in those cells. So we talked a little bit in the first lecture, I believe, about the central dogma. And so that is basically DNA goes to RNA and then goes to protein. So the main thing about DNA is that, number one, it can be replicated, and that's how it's passed on from generation to generation. But also the information in DNA, which A's, G's, C's, and T's are in which position, can then be transcribed into an RNA strand that has very similar, that has the same information, except it's got U's instead of T's. That's not a big deal, though. And so here's like a picture of an RNA molecule. Here's a DNA molecule. The DNA makes this very stable double helix, whereas RNA molecules are usually single-stranded, and they can sort of fold back on each other into interesting things, which we'll talk about later. But this is sort of the message from the DNA, and because that RNA is then translated into a specific amino acid sequence in the protein. So we talked about primary structure of proteins, so the specific sequence that those amino acids are or have in that protein, that's going to be dictated by that message on RNA. So it's often pictured, a good way of picturing it is that DNA is kind of like a recipe book. So if you're working in a kitchen, you'd have that main recipe book basically on how to make each of your different dishes. And then the RNA is sort of like if you just made if the waitress basically puts a ticket in there and says, oh, we need this, this, and this. Then you can go back to that recipe book and start making that specific food. And so the protein would be basically the food that you made. So I hope that metaphor sort of holds up. But DNA to RNA to protein, we'll talk more about this later. But DNA stores genetic information, and then it is transcribed into RNA, which then conveys that information by being translated into a specific protein sequence that's going to dictate the structure and the function of that protein. So the next class of molecules that we're going to talk about are carbohydrates. And so the building block of carbohydrates are monosaccharides, also called sugars. So the words saccharide and sugar are pretty much used interchangeably. So carbohydrates have an interesting sort of chemical structure that's shared between all of them. So they're literally carbon hydrates. So they're basically carbon and H2O. So they have a repeating structure of CH2O. So basically, when you're thinking of carbohydrates, you can think of that it's for the most part just carbon molecules that are associated with oxygen molecules. Now, these oxygen molecules, of course, are electronegative. So they kind of basically, you can think about it as these carbohydrates carry energy through their electrons. So we'll talk a bit more about that, but basically metabolism is taking carbohydrates like glucose and then removing those electrons and using the energy from those electrons to make ATP, which allows your body to function. So there's a lot of different carbohydrates. There's an almost endless supply of different types of carbohydrates. But when we're talking about biochemistry, there's 3 carbohydrates or 3 simple sugars that are most important. And so these are glucose, fructose, and ribose. So glucose is the absolute most important carbohydrate. If you only remember one carbohydrate, just remember glucose. And so here's its structure here. So all 3 of these basically form a cyclic ring, which I'll talk about in a minute. But just look at this CH2O. So this would be a carbon, and then there's a hydrogen on this carbon. So it's CH2O, and then CH2O. CH2O. So again, it's just these carbons are associated with these oxygens that have these electrons that contain energy in them. So glucose is the most important one. And then we have fructose, which you do come across every so often, and then ribose, which we already talked about in terms of DNA and RNA. So those are the structures of those 3 most important carbohydrates. So as I said, carbohydrates are basically just carbons linked to oxygens. So there's really 2 ways that carbon can bond to oxygen to form these carbohydrates. So you can have aldehydes or ketones. And so these aldehydes are carbons that are double bonded to oxygen, then they're double bonded to another group, which we just call an R group, which is usually another carbon or an oxygen. And then it's bound to a hydrogen. So that's an aldehyde. A ketone is similar in that it's carbon double bonded to oxygen, double bonded to an R group, which is usually carbon, but it's also it's also single bonded to another carbon R group, which is also usually carbon. So that's a ketone. Just using this basic rule where these carbohydrates are CH2Os and they're either aldehydes or ketones, you can make simple sugars. So the simplest one that's an aldehyde, so this right here is that aldehyde group, C double bonded to O, bonded to H, and then bonded to something else, in this case a carbon. This is the simplest aldehyde carbohydrate. So it has 3 carbons, all attached to oxygens, and it's called glyceraldehyde. So that would be an aldose because it's an aldehyde sugar molecule. The simplest ketone would be this dihydroxyacetone molecule. So it's a carbon double bonded to an oxygen and then bonded to 2 other groups that aren't hydrogen. In this case, both are carbons. So again, this has 1, 2, 3 carbons all associated with oxygen. So those are the simplest forms of carbohydrates that you can make. So if you build up on this and you start adding carbohydrates, you can get a lot of complexity through these sugars. So you basically just add carbons and then move around where those oxygens are bound to it. So these are all aldehydes, all aldoses. So we have glyceraldehyde. If you add another carbon, you can point this OH group, this OH group, either that direction or that direction, and that gives you erythrose and threose. You can add another carbon to make pentoses, which have 5 carbons. And then if you move around the positions of these oxygen groups here, you can get even more complexity. And so ribose, which we talked about as being important, is one of these pentose molecules. You can also make hexoses. So these are carbohydrates with 6 carbons on them. And so again, you can get diversity by where these OH groups are pointed. So you can have all different combinations to get these different sugars, all of which have names. So glucose right here is a 1, 2, 3, 4, 5, 6 carbon molecule, and each of those carbons are associated with an oxygen molecule. So the first slide I showed carbohydrates as these cyclic molecules. So they form these ring structures. And then I started showing them not as ring structures. So that's because they can basically be formed by these non-ring structures, and then they can form these cyclic rings. So the chemical reactions you don't need to understand really, but really if you take an aldehyde or a ketose and an alcohol group, these can interact to form what's called an hemiacetal. Now, the cool thing about carbohydrates, so if you look at this glucose right here, it has an aldehyde and an alcohol on the same molecule. So these two groups can actually interact with each other, just like what was shown up here. So this oxygen attacks this carbon and forms a bond. And then you can get this cyclic ring. And this group can actually be pointed up or down. That is actually very important, but we'll talk about that in the next lecture. But anyway, so because these two, this alcohol and this aldehyde group, are on the same molecule, they can interact with each other to form this ring shape, which is how we usually show glucose. And just one other caveat on glucose. So why is glucose the most important carbohydrate? Why is that? Why isn't some other carbohydrate so important? Well, this actually has to do with the three-dimensional structure of glucose and how it forms. So I've already shown you a picture of glucose like this. So it's this cyclic ring. It has 1, 2, 3, 4, 5, 6 carbons, all associated with an oxygen molecule. But the way that it's drawn here is just for clarity for the most part. This isn't exactly how it would exist, or not the shape that it would exist in nature. Because remember, all these atoms want to be as far apart from each other as possible. They form this tetrahedral type of structure. And so when you take that tetrahedral or the more stable type of structure into consideration, you find that glucose actually forms this chair form. It sort of almost looks like a chair, like a person was sitting on the chair like this. I can't draw at all. But one interesting thing about glucose, when it forms this chair form, unlike other carbohydrates or 6 carbohydrates with 6 carbons, all of these bulky groups are pointed out to the side away from the body of the carbohydrate. So if they were pointed up, they might interact with each other and be unstable. So because all these OH groups, these electron-rich oxygen atoms are pointed away from the central body of glucose, it makes it really, really stable. And that's why glucose is the most important carbohydrate in nature. And so glucose is so important because of the way that we metabolize molecules. So most of our metabolism, a big chunk of it, is simply taking in glucose and turning it into energy. And so that happens through this complicated process of metabolism. So you don't need to remember any of these details from this slide at all. I'm just demonstrating to you that through biochemistry, we can understand exactly how this molecule, glucose right here, is converted into ATP in this process over here. Now, it's an incredibly complicated process that is catalyzed by enzymes to basically make small changes to the glucose molecule until it's been all 6 of those carbons have been completely oxidized, which basically means those electrons from the oxygens associated with the carbons all that energy's been taken up. So glucose goes through this series of steps where it gets transformed. It gets broken into 2 molecules. Those molecules get transformed, and then you end up with this molecule pyruvate. So this process here is called glycolysis, and it involves a bunch of different enzymes that catalyze these small changes in glucose to eventually get this molecule pyruvate. Now, pyruvate then gets converted into this molecule acetyl-CoA, which is just a 2-carbon molecule. You release CO2 in that process, and acetyl-CoA enters this cycle right here, which you might have heard of, which is called the citric acid cycle, sometimes called the Krebs cycle or the tricarboxylic acid cycle. And again, that molecule is then going to be transformed in different ways, and electrons are going to be taken off of those molecules so that energy can be used elsewhere. And so that energy is actually captured in the form of these molecules right here called FAD and NADH. And what happens to those is they take those electrons and they take it to this complicated process called the electron transport chain, where these proteins that are in the membrane in the mitochondria actually use the energy from the electrons to pump protons through this membrane here. Now, the last step of this is that those protons are used to pump these machines right here in that diagram. So the energy from those protons is used to basically turn that protein like it's a mechanical wheel, and that protein is actually what makes ATP. So again, you do not need to understand anything about this process. I'm just telling you how important the carbohydrate or the simple sugar glucose is, because it powers all of the energy generating processes, so important to life. So glucose is a really energy rich molecule, but it can actually provide even more energy if you took glucose and bonded it to another sugar carbohydrate called fructose. What you get when you do that, you take glucose and attach it to fructose, is you get this molecule here, sucrose. And so sucrose is basically the sugar from fruit. So all these fruits that you eat, an apple, an orange, blackberries, banana, they contain sucrose. So that's glucose plus fructose. And so this sucrose molecule can provide all the energy from glucose, and it can also, this fructose can be modified. So this is fructose right here, fructose, and that's glucose. So fructose can also be modified and metabolized through those processes that we just looked at in the last slide. One interesting thing about humans, and especially other primates and stuff, is that we have co-evolved with plants to develop the ability to see non-green fruits in trees. So we have really good color vision, unlike a lot of other mammals. Like dogs can't really, see these types of colors very well. But us, and as well as monkeys and other primates that live in the trees, we've developed a really good ability to see things like red colors, blue colors, orange colors, yellow colors, so that we can see these fruits, which are sources of sucrose, basically, in the trees. And so this is something that our ability to seek out and crave sucrose is something that is embedded deep within our evolutionary history. And so sugar, basically table sugar, is just sucrose. So if you making a cake or something and you're putting sugar into it, it calls for sugar, whether that's brown sugar, white sugar, anything like that, or refined sugar. Those molecules are just crystallized sucrose molecules. So they're crystallized disaccharides consisting of a glucose attached to a fructose molecule. Now, sugarcane, really the powdered sugar that we all know and are familiar with, was developed from this plant called the sugarcane plant. The technology to sort of cultivate this sugarcane and then to turn that into crystallized sugar that can be transported across long sort of trade networks was developed independently in a few different places. So different types of sugarcane were developed in Indonesia and Papua New Guinea, in southern China, and then also in the Bengal region of India. And so the technology to make powdered sugar, which you can see right here, was developed in India. And so sugarcane has been cultivated as far back as 4000 BCE. And so I thought it was kind of interesting. This is called a sugar loaf here. And this is normally how sugar was sort of transported and put on the table. It's almost like a stick of butter or something. So you would basically just cut off a little bit of sugar from this sugar loaf to put it on your food. But anyway, the development of sugarcane goes back a long time and was developed independently in 3 different places and then spread to other parts of the world. So you can see how that relates to the agricultural revolution and for humans craving for sweet sucrose foods. So sugar became very popular, especially in Europe after for a long time, basically. So it was considered an essential spice that was used and very necessary to make cakes and desserts and also to flavor your tea and everything. So as I said, we're evolutionarily programmed to crave sucrose. However, this led in part of the Atlantic slave trade, which is this terrible thing where basically slaves were imported from Africa to the New World. They developed crops that were then sent to England, and then that whole process sort of repeated. That the slave trade was due in part for the demand for sucrose in European markets. So the cultivation and refinement and production of sugar is a very, very labor intensive and complex process. So you need to plant sugarcane, you need to cultivate it, you need to refine it, and then you also need to pack it, of course, for foreign markets. This is incredibly labor intensive, and sugarcane grows best in these very tropical climates. So not only is that, sweltering hot conditions in which to do this really labor intensive work, but there's also more diseases that were unable to, that Europeans basically had either no immunity to or a high susceptibility to. So the slaves that were brought over basically had a greater resistance to things like malaria and yellow fever. So unfortunately, humans, we obviously crave sucrose, but unfortunately that led to things like the slave trade to demand in part to satisfy the demand for sucrose in European markets. So an interesting thing about sucrose was, of course, sucrose was the main sweetener for soft drinks and beverages like Coca-Cola for many, many years. However, in the 1980s, Coca-Cola introduced a reformulated new Coke. I don't know if most of you are probably too young to remember this. And that was because they had to compete with Pepsi. Pepsi was a bit sweeter than Coke. Coke was more, sort of a mild flavor. And so they introduced this new Coke that was a bit sweeter than the original Coke. Now, this was widely derided. People hated this decision. Fidel Castro, who was the leader of Cuba at the time, actually used it as an excuse, as an argument to talk about, the failure of the American economic system, which is pretty funny because he's also a big Coca-Cola drinker. So this only lasted 10 months. It was a complete failure. And actually, when Coca-Cola announced that they were going to go back to the original and have a classic Coke, Coca-Cola Classic, which would go back to the original formula, the news that night was interrupted to talk about that. Now, some people think that there was a little bit of chicanery going on in the Coca-Cola idea to make new Coke and to switch back to regular Coca-Cola. And part of that is because it was about this time in the mid-'80s that soft drink companies basically switched over from using sucrose to high fructose corn syrup as the sweetener. So this technology had been developed where you could basically take corn starch and then convert it into high fructose corn syrup. Now, it's debatable whether it tastes any different or whether your body likes it more or less than sucrose. That's something we don't really have time to get into right now. But there are some people who think that they just developed new Coke so they can sort of make a switch over from sucrose to high fructose corn syrup. That is not, do not quote me. I don't know if that was exactly what was going through their minds at the time, but it has been discussed. And so because humans crave sucrose so much, we're evolutionarily, designed to crave sucrose. If you have access to this really good molecule that carries a lot of energy in it, there's a reason why feel the need to sort of gorge yourself on cakes and soft drinks and things that taste delicious that have sucrose in them. So unfortunately, of course, if you sucrose is very high in calories. That's why you crave it. However, if it becomes in our distant evolutionary past, it was probably people would come across a grove of trees that actually had ripe fruit, and then you'd be like, I'm eating all of this because I can use that energy, I can store it in fats in my body later. You weren't thinking that consciously, but that's the idea behind eating as much of these sucrose containing foods as possible. However, in modern times, sucrose is widely available, and it's put in sucrose or high fructose corn syrup and are in a ton of different foods. And so this is really calorific, and then it can cause problems in a society like obesity or diabetes from over sort of eating these sucrose molecules. So artificial sweeteners have been tried. They're still trying, and they've tried for many years to develop really good artificial sweeteners that basically activate the same sweet taste receptors on your tongue, but activate them much more than sucrose, give you that same feeling and that same sensation as tasting sugars, but they're not as calorific as sugars. So you can basically have something where only a few milligrams of it will give you the same sort of punch, the sweet punch taste that you would from much, much more of quantity of sucrose. So two of the most famous ones, or three of the most famous ones, are aspartame. So that was one of the early ones. So equal is a form of aspartame. And so that's also found in Diet Coke and all sorts of different drinks. Saccharin is another one. So you can see aspartame is actually, it has like a phenylalanine sort of group here. And then actually looks like glutamate here, or aspartate. So that's aspartame. Anyway, so that's the structure of it. There's also saccharin, which has quite a different structure. And then there's sucralose, which is found in Splenda, which is what Splenda is. And you can see that has more of a carbohydrate type structure to it, but it's chlorinated. So it's got these chlorine groups on the side instead of OH groups. And these artificial sweeteners, they sometimes get a bad rap. Like people are like, oh, aspartame will cause cancer and stuff like that. But in reality, these substances have been tested significantly. There's a ton of research on all these artificial sweeteners that are on the market, and they've been tested for years for their ability to, cause cancers or stuff like that. And the overall consensus is that they're pretty much completely safe to ingest, and they help diminish overall obesity and diabetes in societies, which is a bad problem. Now, another disaccharide which is a connection of two monosaccharides, so two sugars, that you've come in contact with is probably lactose. So lactose is a sugar that's found in milk. So this is glucose right here, linked to another sugar called galactose, which is another 6-carbon chain sugar. So, so this molecule can be processed in your body by this enzyme called lactase. So lactase will basically break this bond, giving you glucose and galactose, which are able to metabolize through those pathways that I showed you a few slides back. So human beings have actually this lactase enzyme. We've actually evolved, certain societies independently across the planet have evolved to basically have this lactase protein expressed throughout adulthood. So in most humans, or at least before this evolutionary switch happened, most humans do produce lactase when they're infants, when they're drinking milk from their mothers. But as you move into adulthood, the lactase enzyme would not be produced anymore. But with the developmental, and so this change correlated with the domestication of cattle, which you can get milk from, or during the agricultural revolution. But this did not occur in all different societies on Earth. And that's evidenced by the fact that 65 to 70% of the planet is lactose intolerant. So they don't produce enough of this lactase enzyme to break down this lactose molecule. And it leads to mostly digestive sort of issues in these people. However, as you can see from this map here, several societies, mostly European and then European Americans and European Australians and New Zealanders, have the ability to retain that lactase activity into adulthood. And that coincides with that cattle domestication during the agricultural revolution. So we've been talking about simple sugars, so monosaccharide carbohydrates, but these can actually form incredibly large polymers that are used for energy storage and structural support. So glucose, again, is by far the most important for making these really large polymers. Glucose makes really large polymers really easily, and this is used for energy storage and structural support. So animals will store excess glucose in the form of glycogen. And so what glycogen is it's really long chains of this sort of structure here. So this is a glucose, and this is a glucose, and they're linked by this bond. And then you can get branches off this bond because this here is another glucose molecules. So this will just keep continuing with glucose, 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 and then you have this branches off, glucose, 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 and then you have branches off here, so more glucose molecules. So these can make really big branched chains. So in animals like us, we store this in the form of glycogen, which is a soluble form of these glucose polymers that we use to access when our blood sugar is too low, or if you're doing strenuous exercise, you're gonna start breaking down this glycogen molecules to provide energy. And then a very similar molecule called starch is found in plants. So starch also has this structure where it's connected in these ways with these branches. But there's some little differences between the two. But and that causes starch to actually be insoluble. So plants don't need to run away from anything. They don't need a storage form of glucose that they need to access very quickly. It's basically they just build up starch during the day in the leaves, and then at night that gets broken down. So it can be actually made soluble and broken down. And the last one I want to mention that's really important, a glucose polymer, is cellulose. So cellulose is shown right here. So cellulose is not an energy storage molecule. It's actually a structural molecule. And so there's a tiny chemical difference between the glucose in cellulose and in starch and glycogen that makes starch and glycogen able to be digested by humans, but cellulose is not. So cellulose is what gives plants their structure. And it's also, if you writing on a piece of paper, that paper is basically made from this molecule cellulose. So cellulose is actually likely the most abundant biomolecule on the planet, just because it's a component of every plant on Earth and gives them their structural support. So we'll talk more about carbohydrate polymers in the next lecture, but I just wanted to show you that these individual monosaccharides, specifically glucose, are able to make these very large polymers that are really important for life. All right, now that brings us to lipids. So lipids, the main component of all lipid molecules are usually fatty acids. So this molecule right here is an example of a fatty acid called palmitate. So these are made from long hydrocarbon chains that end in a hydrophilic carboxylic acid group. So these are polar molecules. So there's a lot of different kinds of fatty acids, but they all share this general structure. So there's this carboxylic acid group here that has a negative charge. And then in this molecule, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 different carbons. And so each of these carbons is going to have, of course, hydrogen molecules attached to it. So one will have 2. This one will have 2 as well. And so these fatty acid chains are basically just made out of these hydrocarbons. So these are non-polar hydrophobic chains, so they don't like being associated with water. Whereas this side right here, that has a charge to it, so it can actually interact with water. So again, there's many different kinds of fatty acids, but they all share this general structure where they have this carboxylic acid group and then a long hydrocarbon chain. And so this is the molecule that we looked at just now. So it's stearate, so it's made of this carboxylic acid group and then this long hydrocarbon chain. And you can notice that there's no double bonds within this fatty acid chain. Now there's different types of fatty acid chains. They can vary, and basically how they vary is in the length of this hydrocarbon chain here. So this hydrocarbon chain contains 16 carbons, but you can have fatty acids with 14 or 12 carbons or 18, basically up to 20 carbons is usually what you would find. Now you can make different molecules by adding double bonds. So this right here is a molecule cis-oleate. So it has 18 carbons, I believe. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So 18 carbon chains, but it also has this double bond right here in basically the middle of the chain. So this causes the chain, it sort of changes the conformation of this hydrocarbon chain. So this double bond in this molecule is in the cis orientation. And that means basically it comes down here, there's a hydrogen here, there's a hydrogen here, and then it goes up in this direction. This actually makes like a kink in the train, or in the chain. So there's also trans fats that have double bonds. So this one, the hydrogens are pointing on this double bond in different directions. So this is where you get the term trans fats, and we'll talk about what that means here in a second. So there's different types of fatty acids that you'll encounter in your day-to-day life. So one thing that you can think about that you encounter all the time is something like olive oil. So olive oil is considered an unsaturated fatty acid. So it's made of a bunch of different fats, but a lot of them are this cis-oleate molecule. So this cis-oleate fatty acids in olive oil, they have this double bond right here. So that puts a kink into the molecule, and that increases the amount of space that these molecules are going to take up. So here's another example here. So there's this kinks in these molecules. And so what that does is it makes a collection of these fatty acids more liquid. So that's why when you have olive oil, it's made up of these fats, but at room temperature, olive oil is of course liquid. And that has to do with these kinks that are introduced by these double bonds in these fatty acid chains. Now, if we take another type of fat that has a bunch of fatty acids, something like bacon fat and other animal fats, those are considered saturated fatty acids because they don't have any double bonds. So these will only take up a very small amount of space, and you can pack more fat molecules into the same amount of space that you would for an unsaturated fatty acid like those found in olive oil. So stuff like bacon fat and other animal fats, they're mostly made up of fatty acids without double bonds, so they can pack together very tightly. And the result of this is that these types of fatty acids have a higher melting temperature. And that's why something like bacon or lard is a solid at room temperature, whereas the olive oil, because it's unsaturated fatty acids with double bonds and those kinks in the tails, those are more liquid at room temperature. So what about these trans fatty acids? So trans fats, you've probably heard that term before. So what this is that basically these cis fatty acids, like you'd see here, that have this double bond and the kink in the chain, these can be converted through a process called hydrogenation into trans fatty acids. So they still have this double bond, but they no longer have the kink in the chain. So this allows something like a vegetable fat that is usually liquid at room temperature. They can interact with each other, and you can pack more chains into a smaller space. And this actually makes these types of trans fatty acids solid at room temperature. So that's really useful if you're trying to make something like margarine, which is shown right here. You're trying to make something that has the consistency of butter, which is an animal fat, which is a lot of saturated fats, from something like olive oil or corn or sunflower oil or corn oil or something like that. So you're basically making these fats that would be liquid at room temperature. Now they're solid at room temperature because you can fit them in more. And so this just has to do with the positions of the different groups on those carbons that share that double bond. So trans fats are really useful for making things like margarine that can be stored and transported way easier than something like olive oil. However, trans fats are linked to things like atherosclerosis, and they mess with the cholesterol metabolism and transport in your body. So there's actually been several negative health consequences linked to these trans fatty acids, and they've actually been outlawed in a lot of different states to sort of combat obesity and heart disease and things like that. So another thing to say about fatty acids and why basically, like I was talking about with sucrose, humans are basically evolutionarily designed to love the taste of fat. I like to say that I've never met a fat molecule that I did not enjoy and enjoy eating. And the reason for that is that fatty acids are really energy rich. So you know that we ingest carbon molecules, and then through metabolism, we sort of, take the energy from the electrons associated with those carbon molecules and turn that into energy in the form of ATP. We've talked a little bit about that. Well, these fatty acids have a lot of different carbon molecules that can be broken down through metabolism. So fatty acids are incredibly energy rich. They're actually more energy rich than glucose and the carbohydrates that you ingest. So, but there's one problem with you. So these fatty acids are going to be, we want them in our body. We want to have them as storage so that we can use them for energy when we need it. The problem is that these fatty acids have this really reactive carboxylic acid group right here. And so that makes them really hard to transport or store in your cells because this carboxylic acid group can interact with other molecules and then create sort of chemical reactions. So these fatty acids aren't really inert molecules that we're able to store and access when needed. So the way that your body has basically compensated for that is that you can take a fatty acid or a couple fatty acids and combine them with this molecule glycerol. And then you get something called a triacyl glycerol or a triacyl glyceride. You might have heard this term before. And that's basically, you take this molecule glycerol, you can see right here, just like right here. And then you can attach 3 fatty acid chains to it. And they can be different. They can have different amounts of double bonds or single bonds, and that'll just change what kind of triacyl glycerol it is. And then this form is not a reactive, so it doesn't carry this negative charge. It's completely inert. So what you can do, this form of fatty acids, you can then store as these lipid droplets in your adipose tissue. So that's tissue that is basically just there to store these really energy-rich fat molecules that you can use when you need to, perform some activity or, make different molecules for your body. You have a really good form of energy in the form of these triacyl glycerols. And so these fat storage molecules are actually what allows humans to survive under starvation conditions. So we talked a little bit about how energy-rich carbohydrates are and that they can form these storage molecules in our body, specifically glycogen, to provide energy. However, that glycogen is really only good for 2 things. So One of those is if your blood sugar level drops way too low, your body can start breaking down glycogen and then taking the glucose from those glycogen polymers and put it back in your blood. A big part of that is that your brain only uses glucose, so it needs glucose in your blood to function. And we'll talk a bit more about that later in another lecture. And the other reason is if you're, like you see a tiger walking down the street and you need to run away very quickly. Your body can really quickly organize that glycogen to run away from that lion or to run that marathon or something. However, glycogen stores run out really quickly. So if you were to exercise basically all out, you'd only have enough glycogen in your cells to last you about an hour. After that, all your energy is going to be provided by these triacylglycerol fat molecules in your adipose tissue, your adipose cells in your adipose tissue. So these fat molecules, because you can store so many of them in a small amount of space and they're so energy rich, this can allow your body to survive for weeks under starvation conditions. So basically you have no food whatsoever, but you can break down the fat stores in your body to provide energy. So if you ever are in an unfortunate situation that some humans have been in, such as the Uruguayan Flight 571, where they basically their plane crashed in the Andes Mountains and they had no access to food. They actually had to resort to cannibalism to stay alive. Your ability to stay alive under those conditions is going to be completely based on the amount of fat stores in your tissue. If you have less fat in your tissues, you are going to survive less long under starvation conditions than the other people in your group. Another situation of this was the Donner Party, which is part of like the westward expansion of the U.S. They got trapped in the winter in the Sierra Nevadas. They were on their way to California. And again, they had no food whatsoever, had to resort to cannibalism. Your survival under these types of conditions is completely going to be due to the amount of fat storage in your body, because the glycogen stores, the glucose stores in your body are not going to, they don't have enough energy, and there's not enough of them to keep you alive under these types of conditions. So another thing that triacylglycerols are really useful for is that they are the basis for soap. So soap is one of the most important molecules or products that humankind has ever made. It allows us to kill, bacteria and clean off dirt on our body and everything. Soap is a huge step forward in terms of human ingenuity. So how soaps are made is basically a reaction of these fatty acid molecules or fats with a basic solution. So in this case, we have a fatty acid molecule. This is a triacylglyceride. If you react that with a highly basic solution like sodium hydroxide, what'll happen is it'll release glycerin and make basically salts of these different fatty acids. So now you have that negative charge back and the positive charge of sodium. So these positive and negative charges can interact with polar molecules when you're trying to clean your hand with soap. And then these fatty acids can interact with and solubilize anything that's more greasy or dirty or hydrophobic on your body. And so that's generally how soap works. So detergents are similar, but they contain a more varied species of lipid, and they're basically usually formed from synthetic sources, as opposed to this fat or soap, where you can basically just, if you remember like the movie Fight Club, they were using fat taken from liposuction, or fat from liposuction clinics, and turning that into soap. But you can also take vegetable fats like olive oil or shortening like animal fat basically, and turn it into soap. So these fatty acids, these triacylglycerols, are the basis for all the soaps and detergents really that you use. So another type of lipid molecule that you've probably come across in your day-to-day life, or I'm sure you have, are waxes. So waxes are lipid molecules that contain these long hydrocarbon chains that are linked by these ester bonds. So this part right here, if you remember from our last discussion, would be like a fatty acid. So one of those, this would be the carboxylic acid part. So how you make waxes is you basically take another hydrocarbon chain and attach it onto one of those oxygens from that carboxylic acid group. So this makes a lot of different types of waxes based on how long those hydrocarbon chains are. So we have beeswax right here. Then we have Carnauba wax, which is a really important wax from plants. And so you can see it has a longer hydrocarbon chain and then a longer hydrocarbon chain. And then spermaceti wax, which we'll talk about in a minute, which actually comes from the head or the melon of sperm whales specifically. And so it has sort of shorter hydrocarbon chains, but they're all linked by these ester bonds right here. So these types of molecules are something that you come in contact with very frequently, and these are also made from these lipid hydrocarbon chains. So the wax spermaceti oil contains that spermaceti wax that we talked about, which is actually called cetyl palmitate. And so sperm whales are actually my favorite animal that on the planet. I find them completely, utterly fascinating. And there's so much that we don't know about them. So this is a picture here of a sperm whale female with her baby. So these are some of the most interesting animals on the planet. They have the largest brain of any animal on the planet. They live very complex lives. They basically only come to the surface to breathe, and then they dive deeper than any other mammal on the planet. There's a lot of evidence that they actually fight and eat giant squid way at the bottom of the ocean. You can see like the sucker marks on these animals, and also you find the beaks, which is like part of the squid, like the squid mouth, in the stomachs of these sperm whales. So they live extremely mysterious lives. And they communicate and hunt and basically see through echolocation. So they have a very sophisticated form of echolocation, just like a bat. So basically they dive so deep that they use sonar to make these clicks that will bounce off of rocks or the topography, and also other animals that they might want to hunt down at the bottom of the ocean. So they use echolocation like a bat basically to communicate. And so their echolocation, so the clicks that they make that come back to them, are actually among the most loud sounds that I think it is the loudest sound that any animal makes on the planet. So if you're a scuba diver or a snorkeling in front of a sperm whale, they can basically make a sound from their snout basically, or their front of their head, that is so loud that it will completely knock you out. And it's been described as feeling like basically a bus just hit you, just from the sound that they make. And so how they make this sound has to do with their very interesting sort of physiology. So they're the only whale that has this really broad head, and it's called a melon. And so what this is it's a very unique organ only found in sperm whales that contains a huge amount of this wax, or sort of a combination of these different waxes, called cetyl palmitate, or actually it's called spermaceti oil, but it contains large amounts of this wax cetyl palmitate. And so they use this. We're not exactly sure why they have this specific organ, but what the thought is, either it has something to do with providing buoyancy or the ability to sink, or it has something to do with their echolocation, which they bounce sounds off and then they're retrieved back into the head. Anyway, so this spermaceti oil that was found that is found in the head of sperm whales, and only sperm whales, has really good qualities for forming candles and other lubricants for machines. So it was very valued in the 1700s and the 1800s, because you can make candles out of it. And remember, this is a pre-electricity time. You can make candles out of it that don't really put off any smoke or have any poor smell at all. this wax and the oil from the sperm whale was also really useful in starting to lubricate machines when the industrial age started. So the demand for this highly prized oil led to an industry that was made sort of famous by the novel Moby Dick by Herman Melville, which was released, I think, in 1851. And so this described the sperm whale industry, where basically these very, very brave men would do this very atrocious act where they would kill very easily. Actually, they're quite easy to kill because they were just coming up for breaths. These sperm whales. And you could get blubber and all sorts of meat and stuff from the sperm whales, but what was most prized was this wax that was in the head of these sperm whales. So that was basically just lifted out of the sperm whale head and put into these huge buckets and then processed on ship. So this really almost led to the depletion of a lot of sperm whale stocks all across the world. This animal might have gone extinct if it continued, but luckily around 1861, so this is a Vanity Fair cartoon from 1961 or 1962, and it's a celebration of sperm whales because they found that there was oils that could in Pennsylvania that had the same properties basically as cetyl palmitate. So that decreased the demand for this spermaceti oil and decreased the amount of whales that were killed each year. So luckily, there's a lot we don't know about sperm whales, but one thing we do know is that their populations seem to have bounced back from that initial sort of hit, which might have led to their extinction. And it's estimated that there could be up to 1 or 2 million sperm whales in the oceans today. Now, another class of lipids that are a little bit unique are the steroids. So these are considered lipids even though they don't have that hydrocarbon chain that we saw in the fatty acids. So that had the hydrocarbon chain terminating in this carboxylic acid group. Instead, steroids are made out of hydrocarbons almost completely, but they form these 4 fused rings that are made of a bunch of different carbons. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So about as many carbons or a comparable amount of carbons as those hydrocarbon chains, but they form these 4 fused rings made out of these hydrocarbons. So arguably the most important steroid in your body is cholesterol. And so cholesterol is kind of a unique steroid. So its main function in your body is to regulate the fluidity of the membranes in your cells. So this part of, and we'll talk a little bit more about this later, but this part of the cholesterol molecule will be embedded in the membrane, and it can actually interact with and push away other lipid molecules to make the membrane more fluid. And then this hydroxide group can actually interact with the polar heads of the lipids in your membrane. So cholesterol is extremely important for regulating the fluidity of your membranes in your body. But cholesterol is also the precursor for a bunch of other steroids that actually have signaling functions in your body. So they actually, when they bind to other cells, these other steroids will actually change the physiology of those cells and then cause a sort of physiological reaction. So the sex hormones, those are all steroids for the most part. So estradiol is a steroid right here. You see it has this specific function that's different from cholesterol. And then testosterone has a similar structure to estrogen, but with some differences. So here's a difference here. Here's a difference here. So that group isn't there. So estrogen and testosterone. Another important one is cortisol. You can see it has a different structure from estradiol or testosterone. So cortisol is actually a stress hormone. So if you're highly stressed, or something like that, it's going to sort of have certain effects in your body to support that. So these are steroids. They're considered lipids, even though they contain 4 fused ring rather than the hydrocarbon chains that we saw in the other fatty acids. And so the last type of lipid molecule that I'm going to talk about are phospholipids. So these phospholipids are really important for your cell membrane formation. So they're very similar to triacylglycerols that we talked about. But so in triacylglycerols, remember there was a glycerol molecule, which is basically like this, and it was attached to 3 hydrocarbon chains. And that's a really good way to store those fatty acids in the form of those hydrocarbon chains. So phospholipids are very similar, and then in that it's basically a glycerol and then 2 hydrocarbon chains, but the 3rd hydrocarbon chain is replaced by this group here, which contains a phosphate and then a charged group. So there's a bunch of different types of phospholipids that all have this same structure. So here, this one up here is called phosphatidylserine. This one's called phosphatidylcholine. And so it has those 2 hydrocarbon chains and then this head group with a positively charged group and a negatively charged phosphate. And then here is another one, phosphatidylethynolamine. Again, it has 2 hydrocarbon chains over here, but then that 3rd position on glycerol is replaced with a negatively charged phosphate group and a positively charged amino group. So these are the types of phospholipids that are in your membrane because they have this very simple structure where they have a hydrophilic head, so a charged head that can interact with water outside or inside the cell, and then these lipid hydrocarbon chains at the end. So you can actually make a shorthand description where it's just this positively charged hydrophilic head, hydrophilic, and then these hydrocarbon tails that are hydrophobic. So these are the phospholipids. And so phospholipids are a really important component of cell membranes for the reasons I just told you. So they have this hydrophilic head and this hydrophobic tail that can be sort of shown here. So hydrophilic head, hydrophobic tail. So what happens when you put a bunch of these phospholipids in water, they will spontaneously form these membranes where these hydrophilic head groups are going to be pointed towards the water outside the cell and the water inside the cell. And then these hydrophobic tails are going to interact with each other away from the water outside the cell and inside the cell. So this is what actually forms. So this structure right here in 3 dimensions is what forms all these different membranes that you find on the outside of the cell. So this would be a picture of the outside of the cell, but there's also membranes around the nucleus and all different little components and organelles within the cell. So phospholipids, because they have this hydrophilic head and this hydrophobic tail, are what are going to form the cell membranes. And we'll talk a bit more about cell membranes in the next lecture. Okay, so that was lecture 3, organic building blocks. So we talked about amino acids, nucleotides, fatty acids, and then simple sugars. So these are the building blocks that are going to make up proteins, nucleic acids, DNA, RNA, lipid molecules, including the membranes in your cells, and also complex carbohydrates that are used for energy and structural support. So I hope you learned a bit. Of course, again, you don't need to memorize any of this information, but do make sure that you get your assignment and your short essay done by the end of the week. But if you do take some time, get to know amino acids, nucleotides, fatty acids, and simple sugars, you'll understand a lot better about the next lecture where we actually talk about proteins, DNA, RNA, and then membrane lipids, and then complex carbohydrates, which are used for energy and structural support in organisms. So I hope you enjoyed the lecture. As always, please let me know if you have any questions.